Hello, my name is Jesson. Today I want to talk about the effect of sediment compaction on macroinfauna in a protected coastal wetland in Taiwan. So before the presenter, I want to introduce the Gourmet Land. Gourmet Land is located in Taiwan. It has Taiwan's largest bullish sail marsh, and it also is the important site for migratory bird. But now it checks for proper visitor regulation. So it keeps the vegetated zone and increasingly degraded. In recent years, increasing ecotourism and visitor have placed growing pressure on the gourmet wetland. So on that case, human trampling is considered a major factor contributed to habitat de- degrad- degradation. Wait. Uh, so I want to tell the purpose of our study. So we want to estimate the link between tramping and sediment compactation and explore how compaction affects macrofaunal distribution and identify species sensitive to compaction. And then most important is we want to purpose management and conservation strategy for the Kome wetland. And now, our study area is Gourmet wetland. The area consists of mastic of silt marsh, mudflat, and sandflat, and it also is a popular ecotourism destination. So we selected total of 35 sites they converting different habitat types and human distribute bands. And we group the site into three categories: low, medium, and high, according to visitor density. At each site, we use a handheld penetrometer to measure sediment penetration resistant to a depth of 10 cm. And then we calculate the average PR, called MPR for each site, and is used to basis for analyze. Then we collect three plate sample at each site, then sorted and count under a microscope, recorded their species and evidence. Then to ensure the sediment physical properties, we collect additional sediment sample to analyze the following physical and chemical properties. And first one is size composition and organic matter content, water content, and bulk density. And here is the result. So we grouped 35 sampling sites into 4 categories based on the sediment compaction. It does low, moderately low, and moderately high and high. And it finds that compaction generally increase with depth. So this figure shows 4 levels of PR values. PR means the sediment compaction. In the depth range 0 to 15 cm. It based on sediment compaction. We choose the low, medium, high, and high as axis. And the down one is the MPR average. And y-axis is distance. It shows and it finds the compaction generally increase with depth. And this figure shows the four level of PR value average in the depth range 0 to 15 cm. it improved that the impact of visitor trampling extend into the midterm sedimentation. You can see also to 15 cm it will increase. We found that the number of visitors in July was significantly higher than April, but the distribution shows two main crowded hotspots with similar pattern in both surveys. This picture is a news that some times crowded are 5 times more than the limit people. And this figure shows, and this figure use contour line to show the mean penetration resistance of each point. And you can see the most outside lines is the lowest PR, and the most inside line is the highest PR, with the most outside line with the space in the line. Yeah. And the MPA has increased with the color in the most MPA at the 19th site. And the SS is longitude, YSS is latitude. And also, these figures use the contour line to show the intensity of visitor compared in these two figures. And in these two figures, we can find April and July has the most crowded in 19 sites. And to compare these two figures, we can find the trampling was closely to sediment compaction. And then we collect total 927 individuals for 23 textiles. The community was donated by MPFOD has 86%, Pilot Kit has 8%, and Biovate has 2%. And in this figure, the SS is TOC, the total organic carbon and sales clay content in these two pictures. And the YSS is MPR. In this picture, I don't know why it's reversed. MPR is in the SS and Y is the Calipolo A. Yeah. And we can see in the sales march, they have the negative relationship for the MPI and TOC and stay content. And in the California A, has negative relationship in any condition. So, most sediment physical parameters shows no significant correlation with PR, but chlorophyll A content negative related with PR. In cell mesh area, PR was inversely related with cells clay content and TOC. So overall, trampling would reduce organic carbon and chlorophyll A with sediment properties, leading to ecological change. Yeah. And this figure shows in the SS representative for level of sediment compaction from each cover, L, median L, and median height and high. And the YSS is the density. The right top is the total macroinfauna density, and this H is for specific species. So we can see the most of the specific couldn't live in the median or high sediment complication levels, and also polychaeta have a big effect of the level. So this figure illustrates the effect of sediment compaction on the community structure of macroinfauna. And so in conclusion, crab were more common in area with higher TOC and more stealth clay, and less common in place with larger grain size. And Biro and coral pond didn't show any clear connection to sediment in our data. And the polychaete, this is a polychaete, appear to be sensitive to sediment compaction than other groups. So we can observe a significant decline in polychaete evidence. So it could be biological indicators. And so in conclusion, we found that when sediment compaction in area with human trampling is at 0.70 MPa is the Gourmet wetland. In the Gourmet wetland, it shows the substantial loss in biodiversity. Sediment compaction in the Gourmet wetland was the lowest at 0 to 3 cm, and it will increase at 3 to 5 cm and also to 15 cm. Resulting sediment may require stronger natural force or active resortation. So here is the way to protect the wetland from this paper. But first, limit the visitor number to reduce trampling-induced compaction. And second, establish a core protection area that remain undisturbed microhabitat, including stage marshes, mudflats, and beaches. Third, create the alternative place and close the original place when compaction exceeds to 0.70 MPa. And fourth, restore moderately to highly compacted areas.
Can you explain these 10 different charts? What does that mean, this data? Because they are talking about different species, but so they can just put them in one figure, but they separate into 10. What's the real reason? In one graph, there are 3 in one, and there are some only 1 in a graph. Oh, okay, thank you.
Can I know, are there any wetland near us?
I just know 鸟松 at the 风山.
Okay, thank you.
Can you explain your purpose again?
Purpose again. So we just want to know the link between trampling and sediment compaction, and know how affect macrofaunal distribution from the sediment compaction, and identify species sensitive to compaction, and find a way to protect this wetland.
Oh, okay.
Yes, I just wanted to know what the relationship between the study and the spatial distribution of visitors.
Spatial distribution will, if people more, it will do more trumps on the mud. Yeah. So distribution of visitor, if the highest, it will be more trump in the land. It could be the highest sediment compaction, equal at the 19th slide, at the slide 19. You can see it is the highest.
Oh, okay, I got it.
This place is a very popular place in Taiwan. So the way it is presented is not ideal. You have a lot of text, but a better way you can introduce this wetland is to show us some nice photos. That would be better, much better than this. I guess maybe at the end, the audience figure out what trampling is. But a good introduction, a good presentation would be to introduce trampling at the beginning of your talk. The term trampling showed up several times, right? I guess that's the most important keywords in the paper and also in your talk. So please introduce trampling. The best way is to show a photo. Again, that would be clear enough. Purpose is good, but I hope that at the end you can conclude the talk by revisit your purpose again. I will say that again later. And there are several pronunciation issues. How do you pronounce this? Mosaic. Mosaic. So it's a pity that you show this at the very late stage. If I were you, I would even use this as my cover. 首页我就会用这个. Such a beautiful place. Here you say that you separate this into low, medium, and high. Here in this map, they didn't divide them into 3 groups, right? There's only number. Okay, so this is a little bit too early, I would say, because I was finding whether they show which one is low, which one is medium, which one is high. So it's a little bit too early. Here you show handheld penetrometer. Please find photo. How do you pronounce this? Macroinfauna. And results? This comes too early. In fact, if I were you, you should show the most fundamental data, which is this one. This is the original reading from the penetrometer, right? That's the original data. So you should show and explain this figure clearly. This is the value from the penetrometer, and this is the depth of the sediment. So these are different PR value, and based on this kind of data, they did cluster analysis, the dendrogram. And based on this, they got this low, median, median, high, and high. So this one would be unnecessary. You don't need to say something that you will explain again with graph. You just explain with graph. Here is confusion. Yes, you told us something about visitors. However, here, there's nothing about visitor. This is the spatial distribution of MPA value. This has nothing to do with visitor. So you should change your header here. And this is visitor. However, here, there are a little bit confusion here because the number here, please repeat again. These are site number. These value, these are distribution of visitor. Their unit is the number of visitor per 30 seconds. And these refer to the contour at the background. You need to say it clearly. Otherwise, there is confusion. Why site 19 has a lot of visitor? We can see that clearly. So if you want to really compare this and this, you should use a third slide to put them together. If I were you, whenever you show this, show photo. Always show photo of animals. I don't think these people, all of them know what is polychaetes. This one is okay, but how do you pronounce this? Chlorophyll A. And what is the meaning of chlorophyll A? Please explain that next time. Print keywords here. What does that mean? Do not just say it's chlorophyll A. Why do you have chlorophyll A in sediment? Explain that. The way it is presented is not ideal. For this is so-called multi-panel figure. To present this, you show one large figure first. Explain it clearly. X-axis, Y-axis, different color bar, different bar. Show one large panel, and then after that, show the whole panel. And later, you want to conclude what kind of animal, what kind of feature they have. Please, this kind of information should be put together with this on the same slide. They should be put next to the data like a polychaetes, show clear the decrease. So after all, because your talk was over 12 minutes, I think after you can stop your data here. For the discussion, please condense it, and then you can even remove it. Remove all the discussion. All you need to do is, after show all the data, come to your purpose and answer each of the point. Then that will make a good talk. Hope to see your improvement next time. Okay, and correct your pronunciation. Okay, good. Thank you, Jesson.